So I really appreciate the opportunity to present this on behalf of my colleagues. Unfortunately, Dr. Menon had a family emergency, so he couldn't make it here, but it was supposed to be a tag team presentation on behalf of Dr. Menon at UTMB, who is an expert in reproductive biology and is in the OBGYN department in the large hospital at the University of Texas Medical Branch in Galveston. Dr. Han, who is a biomedical and electrical computer engineer, who's developing a lot of these chips, and myself, we are at Texas A&M. I'm a toxicologist by training, so that's how we built the team to actually do the qualification. The underlying goal of our Trace MPS Center is to qualify a variety of different MPS for reproductive biology or for women's health. And the biological concept is pretty simple. There is a mother and the fetus. There are two individuals that coexist for a long period of time, and that pregnancy is a very complicated biological and physiological process. And mothers and sometimes fetuses need to be pharmacologically treated for a variety of different conditions, and understanding how drugs not only transfer from mother to a child or could transfer from mother to a child or advertently or inadvertently is very, very important. And while most of the toxicology and regulatory decisions are focused on placental transfer, because this is where the common understanding of drug and chemical transport exists, there is another fetal-maternal interface that is actually the amnion sac around the fetus that also is not without vessels and can also play an important role in both pharmacokinetics and pharmacodynamics. So really understanding, not only how drugs can transfer through the entirety of these fetal-maternal interfaces, but also can potentially impact the fetus is very important. And in our proposal, we put forth 5 different MPS. Some are less complex, some are more complex, and we're taking 1 year at a time to actually do the reproducibility testing between UTMB and Texas A&M, and then also prepare submissions for FDA. So in year 1, we had a 2-chamber crude deciduous organ on chip. So this represents 2 of the cell types. One is the maternal, one is the fetal cell type. The LOI was accepted, as Pasley mentioned, and then in this year we're extending this to 4 different cell types, but still in this non-placental area, and then the placenta chip, and then the combinations will be our out years. So, we started our experiments a little later than some of our other colleagues. So we got going in May 2025. So we're, right into kind of finishing year 2. We finished all the experiments with the 2-chamber chip. We're now working on the 4-chamber chip. We're also preparing our letter of intent and now working on a qualification plan, and had a lot of discussions, while we're not too far away from Galveston and College Station. It is about a 3.5-hour drive, and Texas is just around the corner. So we're trying to get together and meet as often as we can. So I wanted to focus our presentation on kind of context of use, which has been the most important sticking point for most of the iSTAND submissions with the FDA. And as we started building our letter of intent, we really wanted to understand what are the gaps and what are the unmet drug development needs, because really this is where you have to start. It's not that I have a chip and then I start figuring out what are the nails for that hammer. It's you have to start with the nails first and then figure out that your hammer is actually useful. So these are a lot of quotes from the actual draft guidances for the industry from FDA that basically can be summarized in effect to say that during pregnancy, women who are on chronic pharmacotherapy cannot stop that pharmacotherapy just because they're pregnant. So we really need to understand what's happening there. And the flip side of that is that clinical trials usually do not include women who are pregnant and try to exclude women who may become pregnant. So most of the drugs out there have not been tested for women, but are prescribed for chronic conditions. And if some of you guys are here from FDA, SSRIs, are now a hot topic, and a good example of something that, 25% of women are on, and it's unlikely that most of them will not try to get pregnant. So trying to understand the safety or hazard of those types of drugs that are already on the market is very important. Now, these considerations are especially of interest to new modalities, because a lot of small molecules have very similar previous compounds. There's, second, third in line type of pharmaceuticals that we know a lot about. We can read across between. But for new modalities and for especially, first-in-class compounds, these considerations are very, very important. So if you look at the drug safety evaluation that pertains to the FDA, it's really the IND enabling information that is being submitted. It's not really the screening that the companies are doing in-house. And then as you progress through phase 1, 2, and 3 clinical trials, as you progress from healthy individuals to more of the disease population, you are not just completing all of your toxicology right before IND. So for example, developmental and reproductive toxicity studies in animals are actually allowed to be started and completed while the drug is in phase 1 and phase 2 clinical trial. But with a lot of new modalities and a lot of pressure on FDA to speed up approvals, now we're going into phase 1/2, sometimes straight into phase 2 clinical trials. So that type of a timeline is being accelerated quite a bit. But really, these kind of 2 species, both fertility and early fetal development studies and pre and postnatal development studies, are completed on compounds that then will go actually into the population of diseased individuals. They don't have to be completed right before. And this is essentially where we think our type of devices fit. They don't have to be in the pre-IND. They can actually be completed in phase 1 or phase 2 when you actually have a good idea that you have a safe medicine, and now as you're moving into the wider distribution, you need to start getting these data. So obviously, reproductive and fertility and developmental studies are very important, and there is quite extensive guidance. So ICH S5 revision 3 describes very clearly what studies need to be done. Again, these are largely animal studies because we need the pregnancy to be developed, and we need to look both at the fertility and fetal development and actual delivery and post-delivery, kind of postnatal development as well. So this guideline is actually interesting because this is one early example of FDA actually trying to tell developers how to actually do NAMs. Probably was the hardest piece of toxicology to actually use for that, but nonetheless, there is an appendix in this guideline that actually proposes how NAMs should be developed for regulatory use. Unfortunately, not a lot of NAMs have been developed in this space because, again, it's a very hard nut to crack. And industry, is trying to use all methods that are available. And what you see on the right is a recent publication from last year from our industry, pharma colleagues, who have summarized really nicely what in vitro models are available and are being used. But as you can see from this table, most of that use is in the in-house and screening. And they're using mouse and human embryonic stem cell assays and a lot of other assays that are actually quite robust and accessible. But as you can see in the last column, none of these are actually regulatory approved or actually are required by the FDA. So our proposed context of use for this choroidal interface MPS was, again, pretty simple. We propose that it will be used concurrently with preclinical DART studies. So we're not trying to replace animal models. We're basically trying to actually, use these MPS for a slightly different question. Most of the DART studies are really focused on, again, embryo toxicity, on fertility, and on in utero development. Our MPS is really not to test the traditional developmental and reproductive toxicity assays. Our MPS are really more geared towards preventing preterm birth, and that's also a very important phenotype. And placental inflammation and fetal-maternal interface inflammation is one of the accepted causes of preterm birth. And preterm birth has been associated with a lot of the later in life health challenges. So that's a kind of unmet need in our type of a proposal. So we, again, are proposing that the context is to really understand whether a compound can be transferred from maternal deciduo to fetal chorion. Again, it's outside of placenta. There's a lot of PBPK and other models, both in vitro and in vivo models for placental transfer. But again, fetal-maternal interfaces outside of placenta could be an important transfer mechanism as well. And then the second part of this context of use as to whether or not this drug can induce inflammation of that fetal-maternal interface, because it's not the placenta inflammation, it's the fetal-maternal sac that will, if inflamed, will actually push the baby out, because one of the mechanisms of parturition is actually inflammation, at term, and then the baby will come out as a result of that. So the use cases for us are really focused on toxicokinetics, whether there is a transport, whether there is metabolism, and then also, pharmacodynamics. Are there direct effects on those cells? Are there indirect effects, meaning that are maternal cells getting inflamed, producing cytokines, and can actually then affect other parts of the decidua chorion interface. And then really the crux of this is not only having the chip, but also having the cells. And this is where Dr. Menon's lab has spent the last decade developing immortalized human cells, more than a dozen different cell types, because these are very complex interfaces and you have to have human cells and you have to transform them, because you can't just get by with primaries. So we put the letter of intent. It was, again, accepted in mid-November 2025. And as I already mentioned, we proposing this to be done while the compound is already in clinical trials. And with the compound that's already in clinical trials, you have a lot of information. As you can see in the green box, there's a general toxicity studies, both preclinical and clinical. There is a lot of information that you already have. And then in this orange box to the side, you will usually have a DART animal study results. And it's not uncommon that those will be positive, and the pharmaceutical company or the sponsor and the agency will have to think about potentially de-risking, meaning explaining away in a way, these positive results in the animal. And again, there's plenty of examples of where, rat or mouse embryonic development and fetal development is actually different from human, and there are a lot of examples of false positives. So in this type of a process, sponsors and the agency are not expecting a completely clean profile, so there's a variety of strategies to de-risk. One strategy is to terminate development. One strategy is to, deal with the population that this will be used in. Even thalidomide is now on the market and carries a fairly stark warning for women who are of reproductive age not handling that particular compound. So you can deal with, potential developmental reproductive toxicity for an efficacious medicine through a label restriction, or you can conduct additional studies to really understand whether it's true or not. And this is where we propose that our MPS would be useful. And again, it's both evaluation of pharmacokinetics and pharmacodynamics. And then altogether, this information will go into weight of evidence approach. We're not proposing that this particular method will be the only thing that will be used to make a decision. It's never, no data is ever used in isolation in drug safety evaluation. So we've really appreciated the time and the effort that our colleagues at FDA have spent in reviewing our proposal. And we probably learned as much from this process and from the feedback that they give us as we actually learned ourselves in actually putting this together. So feedback has been tremendously instructive. They've asked us a lot of technical questions to explain. They suggested maybe expanding the context of use. They recommended a number of model validation steps, and also they challenged us to do a better job and perhaps putting more context into that weight of evidence evaluation. How exactly would you decide, and would you have a threshold or some sort of a continuum in your phenotypes from this particular chip? So we're busy putting together a qualification plan, and at least in our team, we have all the protocols for how this can be made, how the experiments should be done. We understand the robustness and reproducibility. It's really the positive and negative compounds that will give future users confidence. So selecting those for non-liver applications is challenging, because fortunately for liver, as we'll hear from Dr. Tong and others, there's a lot of effort on drug-induced liver injury, on DILI positive, DILI negative compounds. There are lists and lists, and there are tons of studies that have used those lists. In this type of space, we really are, trying to make our own decisions. And again, our plan is to go back to this guideline, the DART guideline. They have a list of positive compounds. Again, they're positive for fetal, and developmental endpoints that are not really positive for preterm birth, but we have gone through that list and selected a number of compounds. So we think that the positive controls, a subset of those compounds actually can be traced from the rodent pregnancy to human pregnancy in terms of preterm birth. So they will be used as positive controls. And then we also have a number of negative controls, both from the guideline and also other clinically approved drugs. So in summary, we, again, are busy at work. We appreciate all the feedback, and we appreciate any feedback we can receive at this meeting as well. So thank you so much.